You have to look on there and track where oxygen is where it's showing up. Why is that happening? Go ahead. Yeah, absolutely. And that's an easy thing for a lot of people to just kind of miss that idea. And so like I've had students in the past, when I ask a little bit about that on the exam, they're not linking those two ideas. The reason oxygen is being given off is that water being used as an electron source. And if you strip electrons off water, you will get oxygen as one of the products. Okay. So oxygen, water, H2O, those are two protons, the oxygen molecule. And so the plant is trying to get electrons from water. Water is extremely stable, by the way. It's a pretty amazing process that the plants can pull this off. And so, but they do with the help of light energy, and they're able to extract electrons and send that into the electron transport chain process. And then they also really like having those protons as one of the products because that helps build a proton gradient. And then oxygen is also formed, which we really like because it allowed us to do this on this planet. Okay. So, and when I teach about this in general microbiology, I really make a point of this because not all organisms that do photosynthesis will use water. And so not all of them will actually generate oxygen. We're so used to thinking about photosynthesis, oh, it produces oxygen, but that's not always the case, just so you know. But with plants, they are certainly doing it in this manner, using water as their electron source. Okay. So we see that this is an electron transport chain process where we have the electrons going from donor to acceptor on down the line. You have these big transmembrane complexes, the photosystem 1, cytochrome B6F, photosystem 2 on the far left, cytochrome B6F, photosystem 1, and then the ATP synthase. All very huge and pretty much fixed in position. So you have to have these little mobile carriers to help move electrons in the chain process. And so you have the plastoquinone doing a role here, the plastocyanin. And so basically I need you to appreciate that you have these big complexes, but you also have these mobile carriers that will allow the electron flow to happen pretty seamlessly on down the line, so to speak, until you end up with the final or terminal electron acceptor of NADP plus sitting there waiting for the electron. Once it gets that, we say it's reduced to the NADPH. And that's one of the huge goals for the plant because they can use that NADPH. NADPH is a very good reducing molecule or reducing agent, which means it likes to, in this case, float around in the stroma and give off electrons where it's needed. That's what it's going to do next. And so we'll talk about that. So all of that electron flow is cooking along all day long as the plant is absorbing light energy. This is going to happen, which also allows for that proton gradient. Okay, so hand in hand, you can't separate these two ideas. If you have that electron flow, you're going to get a proton gradient through this process. That's what they're trying to show here, because when you strip electrons off water, you get protons. That's awesome for the plant. And that's what they're showing down here in the lumen area. And then you're also seeing protons essentially disappearing from the stroma and ending up in the lumen. So you start to get this shift in an accumulation of protons on the lumen side of the thylakoid membrane, and then less on the stroma side of the thylakoid membrane. So then by the laws of diffusion, those protons want to go and even out their concentration. So they're going to want to cross that membrane probably to the stroma. Can't go through the membrane because the charge won't work. So they have to go through that ATP synthase. And as they do that, that flow of protons actually causes a rotational torque force between subunits. allows for ADP and inorganic phosphate to react with each other. And then you end up with that lovely ATP. So that's a huge second payoff for the plant. Get that NADPH, get that ATP. Oxygen, they're really not too concerned about for what they're trying to do here. That's not really their goal. We love it because we can breathe, survive. But for them, that's not really what they're focused on. They want to get NADPH and ATP. Questions on the light reaction idea? Clear as mud? Spend some time with it, please. Now remember, this content's not going to be on Friday's exam. So you have some time to think through this, and I really want you to think through this. This is an amazing process. It's just phenomenal that plants can do this. So please spend some time learning about this. Well, okay, so we're still moving into the chloroplast here. We have the stroma portion, that's just the watery portion. If you crack open a chloroplast and look inside, all that watery stuff is called the stroma. And that's great that the NADPH and ATP are being made right there, because then the next step of photosynthesis takes place right there. So it's very convenient, very efficient. So we're zooming into the stroma again, and we're thinking about the Calvin cycle. And so once they figured out that CO2 is reacting with a 5-carbon sugar, things kind of quickly fell into place after that. That took a long time of figuring out. And so here you're looking at the idea of CO2. So the stomata, right? The stomata of the plant have to be open. They have to take in that CO2 and react it with RUBP, that 5-carbon sugar. And then you get this 6-carbon really quick, it's a short-lived molecule, very unstable. And then that breaks into 2 identical molecules of phosphoglycerate. And the 3 on there just means that it's the 3rd carbon that has the phosphate on it. But they're identical molecules of another way to say phosphoglycerate, by the way, is phosphoglyceric acid. That's where the PGA, the A, comes from. It's phosphoglyceric acid. Okay. So as long as the plant has its stomata open so it can get a source of CO2, and as long as it has RUBP, this is just going to happen. If those 2 molecules come in proximity to each other with the enzyme, it's going to work. Let's talk a little bit about that. Well, no, let me finish the thought here. All right, so we have the PGA formed, and then you see the PGA, those 2 molecules will go through a series of reactions, and you'll finally end up with the G3P being formed. Okay, so that's kind of like the second phase of the Calvin cycle. Now this needs a little help. Here you see ATP is coming into play from the light reaction. So is the NADPH. So both of those molecules need to be part of this process to convert PGA to G3P. And then G3P, that's an important product, because if enough G3P builds up through running the Calvin cycle, if you get enough of those, then you can take some of them away and put them into a separate pathway to make sucrose. Right? So the Calvin cycle is not directly, like nowhere in the Calvin cycle do you see sucrose showing up. Okay, so the Calvin cycle job is really just to make sure that you're taking CO2 in and reacting it with RUBP. Essentially, you want to get that G3P building up so that you can take some of that off and put it into a separate pathway. So this green box is alluding to that idea that there's something else going on here and they're just not showing it. Okay, so some of that G3P gets sent over into another pathway, and then your sucrose will get made. And that's the sugar the plants really want, is sucrose. Okay, so that's great. But you need to keep the Calvin cycle going to get enough G3P to make sucrose. And so you got to keep going until you finish the cycle. You got to come back to the beginning. And so this is the idea of taking that G3P molecule, and then you see this arrow, this section right here. There's a whole lot going on there, which we're not going to get into in any significant detail in this class, but we do in Plant Phys. Lots of reactions going on there, but the end result is that these reactions will end up with this molecule. You'll get an accumulation of this ribulose 5-phosphate. So it's pretty close to ribulose 1,5-bisphosphate. Not quite there. It needs one more phosphate, right? And where do you typically get a phosphate from? Well, ATP is usually a favorite source. And so you have to phosphorylate this molecule with ATP so that you can have 2 phosphates. And so that's where the bis comes from. Ribulose 1,5-bisphosphate means you have 2 phosphates. Okay. So the Calvin cycle, or C3 cycle, has to keep going round and round and around and around all day long. But it's linked to another pathway that gets you sucrose. Okay? And so that's what the green box is. So you got this, look it long enough, and they'll take some of that G3P off and push it into a different pathway that will allow for sucrose. And then once the, normally this is happening in the leaf tissue where all the chlorophyll is, chloroplast. And so once the leaves start accumulating enough sugar, then eventually they need to share some of that because like roots, typical roots, they can't do this. So they're like, I need sugar. And so the plant has to share that. We'll talk about how that happens. But if you think about the leaf tissue, they're doing this as soon as the sun comes up and strikes their body, they kick into this immediately. And if everything goes okay, it'll go all day long. And some of that G3P keeps getting put into sucrose synthesis all day long. Now, the enzyme that is probably the heaviest studied enzyme in the plant world is this one right here, Rubisco. So Rubisco is the enzyme that allows for that very first step of the Calvin cycle, taking that 5-carbon RUBP and reacting CO2 with it. So Rubisco, as long as it takes CO2 into its active site and RUBP into its active site, boom, it's just going to happen. Those will react and you'll get your PGA eventually. Well, this is a very unusual enzyme. There's only a handful of enzymes that are similar to this. So, do I have it? Okay. Rubisco is an acronym. It stands for that RUB part of it is referring to the ribulose, essentially rubus. The rubus portion stands for the ribulose 1,5-bisphosphate. Okay, that 5-carbon sugar idea. And then the C stands for carboxylase. And the O on the end stands for oxygenase. So it's a huge mouthful when you say Rubisco full out instead of using the acronym. Rubisco is ribulose 1,5-bisphosphate carboxylase oxygenase. That's a huge mouthful. So now you know why they do an acronym. Thank you. And so, but having said that, it's important that you distinguish Rubisco is the enzyme. And the way I remember is as long as I know CO on the end, as long as I remember what that stands for, it stands for carboxylase oxygenase. Anytime you see ASE on the end of a word in science, it means it's an enzyme. Okay? That's the way I remember it. Now, I'm making a big deal about this because I've seen in previous semesters, students confuse RUBP with Rubisco. Very different molecules. Okay, RUBP is a 5-carbon sugar. Rubisco is the enzyme that will work with that sugar, but it's an enzyme. It's a protein. It's not a sugar. So please start getting that straight in your head so that when it comes time for quizzes and tests, you aren't mixing those up. Now, why do they call it a carboxylase oxygenase? Why do they stick that on to the acronym? It's because this enzyme is kind of funky. It can take CO2 into its active site and react that with RUBP, or it can take oxygen into its active site and react that with RUBP. And to Rubisco, one is just as good as the other. It doesn't really care. It's very egalitarian. Yeah, I'll take CO2 in. Oh, there's oxygen. Yeah, sure, I'll take that in. And so we say it's a competitive reaction process. Whatever is in higher concentration around Rubisco, if CO2, if there's more of that around Rubisco, it wins, comes into the active site and reacts with our RUBP. However, if oxygen, if there's plenty of that around, well, that wins the game. It's going to come into the active site of Rubisco. And so Rubisco can't really tell the difference, so to speak, between CO2 and O2. It's the fancy way to say it is its binding affinity is low. It doesn't really have a strict, I'm only going to bind with CO2. It doesn't do that. It's pretty like open, like, oh yeah, I'll take oxygen, that's fine. So we say it's a competitive reaction. So either CO2 is going to react with RUBP or oxygen will react with RUBP, the 5-carbon sugar. Well, that causes some problems for the plant because if oxygen wins and comes into the active site, we end up with a funky reaction. So what happens is this bullet point here where you see RUBP and oxygen reacting. And you do get a PGA molecule, which is great because that's what the plant wants. But the other problem here is, yeah, you only get one PGA instead of 2. And so you get one PGA and then you get this funky other product called phosphoglycolate. This is a 2-carbon molecule that does not fit into the C3 cycle. Like it doesn't work. There's no enzymes to work with it. And so it's like, okay, that's a loss. It can't fit into the Calvin cycle. Instead, it has to fit into a different cycle called the PCO cycle. And I'll show you that when it goes through the PCO cycle, this 2-carbon phosphoglycolate, the plant can end up getting something from it. It can gain a little bit from it, but it's still not the best situation. It's actually referred to, this PCO cycle, as photorespiration. So you're respiring right now. We're all doing it. And when we exhale, we're giving off CO2, right? It's lost to us. It's out of our body. That's what they're trying to say here with the idea of photorespiration. They're saying that the plant actually loses CO2 out of its body in this process, rather than taking CO2 and making sugars from it. So it's doing the exact opposite of what you're trying to get the plant to do with the C3 cycle and all of that, the Calvin cycle. So it's a loss. It's a loss of carbon. And so they'll say it that way too. It's a loss of carbon. What they mean is the plant is actually going to lose CO2 and it won't get fixed into sugars. So now you have to think back when Rubisco was, formed through the process of evolution and natural selection. This happened actually before plants came about. Microbes have Rubisco, and microbes were the first organisms on this planet. And so we're talking like they think life cells arose on this planet like 3.5 to 3.8 billion years ago. And so Rubisco probably, let's just say 3 billion years ago, we start seeing the idea of Rubisco. Back then, there wasn't any oxygen in our atmosphere. So if you put it into context of the evolutionary process, at first glance, you'd be like, well, why would the plant be so stupid to have this sloppy enzyme? That's just silly. But when you think about it, Rubisco evolved during a time period on Earth where oxygen wasn't a thing. And so it didn't matter that it was floppy back then. It didn't ever see oxygen, so it didn't matter. So that's one big reason why they point to the fact that this enzyme is a little bit, sloppy about what it will take into its active site. Let's look at a flow chart to try to get this in context here. So here on the very top, you see the 5-carbon sugar, REBP. And they're saying, let's go off to the right and talk about, it's weird. It doesn't want to erase. Okay, let's go to off to the right. High CO2, low oxygen. So if the sun comes up, strikes the plant body, it opens its stomata, starts taking lots of CO2 in under those conditions, it's going to run the Calvin cycle, no problem. Rubisco's going to see CO2, 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 and we'll react that with RUBP, and you'll get those 2 molecules of PGA. Calvin cycle cooks along, no problem. Off to the left is where things can go wonky and get a little troublesome for the plant. If you have low CO2 levels around Rubisco, which means, so if Rubisco is inside the plant and it's not seeing very much CO2, what's going on? What's the plant doing that's preventing Rubisco from seeing CO2? What's going on? Yeah. Yep. The plant's going, closing my stomata. Why would a plant want to close its stomata? Under what conditions would it be prudent to do so? Drought. Nighttime. Yeah. So if, let's say it is, what time is sunrise these days? 8:30, 8:15, something like that. Let's say it's 8:15, sun comes up, and we're not in the middle of winter, and the plant starts going, okay, yeah, I'm going to open my stomata, and then it goes with its roots. Uh-oh, I don't have enough water. Maybe this isn't such a good idea. So then let's say it closes down its stomata, conserve water. So now Rubisco isn't seeing much CO2, and as we go along through the next hour or so of that happening, it's going to see even less and less and less and less. So then you're going to see Rubisco is going to be surrounded by low CO2, but there's still oxygen in its cells. It's doing photosynthesis, right? So oxygen is being generated. So the plant's busy doing the light reaction. There's plenty of oxygen showing up in those cells. But because the stomata are closed, the CO2 is crashing. So then the plant is forced to do the PCO cycle because Rubisco is like, yeah, if there's more oxygen around me, I'm all right with that. I'll take that in. It's a competitive reaction. Oxygen comes in, reacts with REBP, and you only get 1 PGA, and that can feed into the Calvin cycle. So you can get the Calvin cycle to go. It's just really not very high level of it going. And then that phosphoglycolate is the other product that's really the problem. It's like, now what are we going to do with that? It's a 2-carbon molecule. There's nowhere in the Calvin cycle to fit that. I don't have any enzymes to work with it in the Calvin cycle. And then you see this arrow, arrow, arrow. And so what they're trying to show here is there's a whole lot going on here in order to work with that phosphoglycolate. And they're showing that CO2 is actually one of the products, and that's the photorespiration idea. It's like breathed out, so to speak, by the plant, given up by the plant. So that's it. So the reason I want to talk to you all about this idea is that in this class in particular is this understanding this dilemma helps you to figure out why certain plants live where they do in the world. Like, have you ever wondered why, some plants, why can't we grow, coconut trees up here? Why can't we do that? Why do they only grow in the tropical areas? And so thinking about where plants have evolved, what their native habitat is, and if you had the good fortune to travel a lot and you've seen different plants being very healthy and growing in these different landscapes that, there's no way they would survive up here. It's primarily because of this idea that we need to think about why they're so successful in certain areas of the world and they can't grow in other areas, right? And so here we're looking at this idea of rubisco. Rubisco is quite sloppy, and yeah, it can take CO2 or it can take O2 into its active site. So here, if you're a visual person, sometimes it helps to think about what's really going on. You have that 5-carbon sugar, RUBP, and it, for some reason, probably the stomata have been closed for a while, so oxygen is building up. So that's what rubisco works with. And you get that PGA on the bottom here. That's great. That can go right into the Calvin cycle. But this phosphoglycolate, you see, it's a 2-carbon molecule. There's no enzymes in the Calvin cycle for that one. And so that gets shunted into the PCO. That's the PCO. So that arrow that they were alluding to, there's a whole lot going on here. Now, I'm not going to ask you to know the PCO cycle in detail, but there are some things you do need to know. You need to know the very start of it. Okay, so both of these are the PCO cycle. I put 2 images side by side. They're both the same thing. It depends on how your brain works, how you want to study off of this. Both are saying this pretty much the same thing though. So let me see if I can work with this one. Okay. So over to the left, I'm in the high left there, you see the ribulose 1,5-bisphosphate and oxygen reacting. So that's where rubisco is. And you see one of the products is phosphoglycerate off to the right. And then I'm saying, yep, that can go right into the Calvin cycle. No, but then the other product, like so now that's what's entering into the PCO cycle. So know what kicks it off. That's really critical. And then understand that once the PCO cycle, once phosphoglycolate goes into it, we have 3 different organelles. I need you to know it starts in the chloroplast, and then it has to move to the peroxisome organelle, then to the mitochondria. So we got some complexity here. You're talking about shunting these molecules between organelles. That's complicated. And then you're going to see that it circles back around where this reaction pathway comes down into the mitochondria, so to speak. And then you can see in the mitochondria, I wish the artist had been a little bit more generous with color or something, because in the mitochondria, you see CO2 being given off. That's the respiration idea, the photorespiration that we alluded to. That's a big deal. Okay, so this is considered a major problem. The fact that the CO2 in the mitochondria is being given off, and that is what is lost. That won't get fixed into sugars. So that's the photorespiration idea. So that's a loss of carbon. That's another way they say it. It's a loss of carbon. Then you're going to see that it circles back out of the mitochondria, the products go into the peroxisome, and then we finally work our way back to the chloroplast. By the time the PCO cycle is all done, you end up taking that phosphoglycolate through that whole process, and actually have to do it twice because phosphoglycolate is a 2-carbon, you need to end up with a 3-carbon. So the way it works, do the carbon counting on this diagram, you convince yourself you have to run this twice in order to convert the phosphoglycolate to phosphoglycerate. And once that happens, once you get that other phosphoglycerate, yeah, go ahead and put that into the Calvin cycle. That works. Complicated. Lots of resources needed. CO2 is lost. ATP is used. not really a good idea. Now, the plant evolved this to recapture some of that carbon. It is, getting some back. It is creating a phosphoglycerate from this mess. So there's a payoff, but there's also a loss. So when they do head-to-head experiments where they force a plant to do the PCO, they create conditions where we're going to make you do the PCO. We're going to make you do the PCO. And you do that too often to the plant suffers. Its growth slows down. Its yield at the end of, if you wanted to harvest it, the harvestable yield is much lower. And so in general, plant physiologists say, yeah, really not a good idea to have photorespiration or this PCO cycle happening on a regular basis. And plants came to that conclusion too, because they've evolved mechanisms to try to minimize this, which we'll have to get into next week. Okay? But they have figured out through the process of evolution and natural selection, they have figured out ways to minimize this. But it really depends where they live, how they handle this. And so we'll get into that next week. Okay, so to be continued on that idea. Pretty amazing process. Okay, so now let's shift to the idea of the exam. So I posted a study guide in the PowerPoint slide section of EduCat. It's there, a Word document. I'm going to stop share for a second here. Okay. All right, let me get out of this. That. Okay, so chapters 1, 3, 4, 11, and 14. So chapter 1, I didn't really talk about. There's just a handful of ideas in there to think about. Pretty straightforward. And so really we jumped into plant cells and tissues. So let me just pull up that slide set and see if there's any lingering questions that need to be addressed. I'm going to duplicate. Okay. See if I can share so the recording would pick up. Okay. All right, so going through your notes on plant cells and tissues. So you were asked some questions about this on the lab exam or homework that you did. So I tend to focus in obviously on things that are pretty unique to the plant. So I don't, I'm not going to really bother with some of the organelles that we have, but some of the unique organelles that plants have evolved are more important that I'll ask questions about. So what are some of those organelles that you think I might ask questions about? Yes, chloroplasts. Ding, ding, ding. Yes. There's another one too that's pretty important for the plant because it doesn't have vacuoles, that huge central vacuole. Believe it or not, that's super important for the plant because it doesn't have that internal skeletal system like we do. So that when a plant cell is mature, the largest organelle is the vacuole for sure. And they like to pack it full of water so they can create that turgor or water pressure against the cell wall so that whole tissue stays nice and turgid to fight gravity. The leaves can stay nice and flat so that they can capture all that sun. Cell wall structure, pretty important. We don't have cell walls. Plants have to do that for a lot of that has to do with structural support, protection. So knowing the details of cell wall structure as provided in the PowerPoints here. What happens when a plant cell puts down a secondary cell wall? It dies. Is that a bad thing? No, that can be very good, actually. Makes water transport through the plant body super efficient. by the way, water transport through the body, that's vascular tissue. Which kind of vascular tissue does water transport? Xylem, very good. So xylem does the water transport, phloem does the sugar transport. So knowing the basics of that. For sure, I'll be asking about vascular tissue and the different types of vascular tissue because we'll talk about that for the rest of the semester. Vascular tissue comes into play in so many situations we'll talk about and what you'll do in lab as well. Questions? Yes. Correct.
But then it, like, can you just kind of explain to me then, because it says the tissues produced by meristems, simple tissues, like above.
Where?
If you just went back to where you were for the parenchyma cells for 24.
Mm-hmm. Yes. So tissues produced by meristem cells. So meristems, we don't call them parenchyma cells. They're meristem cells. And they're like, think of them as, what's the best way to describe them? They're like proto cells in a way. They're or baby cells. they're not quite anything. They're, you know, like they're not a parenchyma, they're not a collenchyma, they're not that. They're like embryo cells. Think of them that way. Yeah, stem cells. Kind of that way. I think if you use that as an analogy, that might help. Other questions on this slide set, content in this slide set? Yes. Possibly there might be a visual, you know, so I think what maybe you're envisioning is like slide 11, maybe, you know, like what's a primary cell wall? What's a secondary cell wall? What's the plasma model? You know, what's the cell, cell? So yeah, there might be something like that where I'd ask you, but it'd probably be like a drag and drop where you, the terms would be there and you drag them to the figure. I've done that before, yeah. Not too much. It's like cement. Think of it like cement. It helps stick the cells together. So yeah, the middle lamella has a lot of pectin in it, and pectin is nice and gooey and sticky, and it really does help keep the cells stuck together. Other questions? Ah, yeah. So vascular cambium. Are you looking at a particular slide? I got to stop share. And let me reshare. Okay. Which, did you have a slide number? Vascular cambium, is that what you were asking about? So think of the vascular cambium and cork cambium. They're kind of like two distinct layers. The vascular cambium is the ring of cells in between the swath of kind of grayish pink. That's all xylem, right? And then this stuff, the darker pink off the perimeter, that's primarily phloem. And so the ring of cells in between those two is the vascular cambium. And the vascular cambium is super important because, so it's, my pen is red, so it's not gonna show up very well, but it's that ring of cells that goes all the way around. And when the vascular cambium is doing its job correctly, what it's doing is going, okay, I'm gonna divide. I'm gonna produce a xylem cell to the inside, and I'm gonna produce a phloem cell to the outside. And so that whole ring of cells is doing that. Xylem cell, when I divide, produce two cells. One will be a xylem cell, one will be a phloem cell. And that allows for building xylem and phloem, which also increases over time the stem, the tree trunk diameter. So that's the vascular cambium. Cork cambium is, I don't know if you can see that ring of dark, like that dark ring here. That goes all the way around again. So it's further out, and it's going to, the cork cambium is going to produce bark. Okay, bark tissue, which is primarily for protection. And not all plants, by the way, not all plants have a vascular cambium. We'll get into that. So we're only talking about certain types of plants, like, you know, the beautiful trees we have in this region. They all have vascular cambium. But like corn plants, they don't do that. You never see a corn stem get massive. All right, so there's a lot of plants that don't have this. Other questions? We're getting low on time here. What about plant growth? Tropisms, thigmotropism, nyctinasty, all those terms, and what does that mean? I'll be asking about hormones, auxins and cytokinins, hugely important, but the other three we mentioned, don't forget about them. What does auxin do? You know, the whole phototropism idea, going to the dark side. So the details of auxin in particular, you need to make sure that you've covered that in your studying. But essentially knowing, you know, a good way to do it is kind of like have the PowerPoint out and kind of do a compare and contrast maybe when you're studying, so you can keep straight which hormones do what, because I'll be asking like, so if I ask about a hormone's function, what I tend to do is I will say, if I want you to identify a hormone, I'll say this hormone does this, this, and this for the plant. So I don't say this hormone does this. What is it? I give you more than one clue. You know, I'll say this hormone can do this, this, and this. Which one is it? Okay, so maybe doing the compare and contrast as you're studying so you can keep track of what each hormone can do for the plant. And just be aware, what I have in the slide set is not in totality what these hormones do. I'm just giving you a taste of what they do. Okay, so I'm limiting here on, believe it or not, this is a limited idea of what these hormones can do. So hopefully you saw some of those videos when you were studying. Did anyone watch to the very end of that one video where I'd say, watch to the very end? You should do it. It's in one of these slides where I say, watch it to the very end. It's humorous. You'll get a laugh. Oh, phytochrome. Gotta know that. How are we doing on that? That one's a little trickier. So phytochrome, the P in the abbreviation, you know, the P sub R, P sub FR. What does the P stand for? Really? What does the P stand for? Stands for phytochrome. Okay. Stands for phytochrome. And then the R is what quality of light is it designed to absorb. So P sub R means it's the phytochrome pigment form that can absorb red light. And when it does that, it will shift to the phytochrome form that happens to be really good at absorbing far red light. And it can keep toggling back and forth. And so it's actually physically changing shape. If you want to think of it that the P sub R is the phytochrome, like to me it's like the letter M. You know, it's the M form of the phytochrome. And then when it gets, when red light is Well, that's the piece of FR. So they're both, the P is, it's phytochrome. It's just different shapes of it. And that allows it to absorb different qualities of light. Red or far red. So try to get that straight in your head. I think this diagram goes a long way to helping with that idea. In that it's the P, that's all phytochrome. That's what we're thinking about. It's just which shape are we thinking about? Other, any other concerns about phytochrome? I see a hand somewhere. So what it can do for the plant, what it allows the plant to assess. And it's really, remember, it's the ratio of these two really that the plant's constantly assessing. How much piece of FR do I have in my body? How much piece of R do I have in my body? And that's going to tell the plant, am I in light? Am I in dark? Am I getting shaded? What should I do about that? Is it time to flower? So today is Wednesday, right? So I check my email quite frequently leading up to exam day. So if you're studying and you're like, I don't know, shoot me a quick email. And try to respond to you. Even into the evening, I check emails on these types of leading into exam day. Okay. And that Word document is posted in EduCat by the PowerPoint slides. It's just getting you to think about what to focus on. Okay. All right, I guess we gotta go. Yep.
It's just a barrier.
Yep, it's a ring of, it's a meristematic ring of cells. So it's meristematic, which is great because it can constantly divide and produce biome to the inside. Or to the outside. Yep. All right. See you later.